It's truly a pleasure and an honor to be here. And so let's get started. So I have no conflicts to disclose. So this slide right here is a new one. It's one that I don't normally show, and it's actually one that I felt was important to give a more general overview of the various genetic causes of male infertility, despite the fact that where I typically spend most of my time is in the intrinsic testicular. Ever since I started working in the field of male reproduction 15 years ago, I've been primarily focused on spermatogenesis, germ cell production, all of the genes that are required for the many different steps. So there's many genes, reproductive tract specific, in fact, that are required for fertility in men, often shared with women, and they can all be found in infertile men, depending on their phenotype. But there's also pre-testicular causes and post-testicular causes, and if I were to give a comprehensive overview of the causes, then I would go into those as well. So for pre-testicular causes, there's Kallmann syndrome, which basically consists of degeneration of the gonadotropin-releasing hormone-producing neurons, which is also linked to the neurons that are required for development of olfaction. So it's interesting, some of the men who have these defects also lack smell. There's a number of genes that can cause that, and the number one phenotypic characteristic would be lack of puberty, lack of, it's a hypogonadotropic hypogonadism-related disorder. And so then the other two that are listed here, they're both pretty much similar, and what they consist of is a defect in red blood cells and hemoglobin, and the genes that are required for proper hemoglobin, they're disrupted, and therefore what happens is there's an iron overload in various tissues that include the hypothalamus, pituitary, and the testis of men, which can then result in infertility, right? So then for intrinsic testicular, I put monogenic causes all the way at the bottom, even though it's a huge source of variants that can cause disease. But the other ones that are also important is the chromosomal abnormalities, like Klinefelter syndrome, which is a XXY. These men are perfectly normal otherwise, except they realize that they're azoospermic eventually. And so that can create issues. Sometimes they have other metabolic disorders, autoimmune disorders, but they could go most of their life not knowing that they have this unless they're tested. And typically the test that's ordered is a karyotype test. And then the other tests, karyotype test would also detect 46XX male syndrome, and so how could an individual present phenotypically as male but be XX? And so the only way that could happen is if there's SRY gene somewhere. So if the Y is completely gone, then it must have ended up somewhere else, right? And so that's important to note, and I'll show an example where I found something similar to this, but it wasn't XX specifically. And so other chromosomal disorders, Robertsonian translocations, transversions, duplications, and I cannot talk about genetic causes of infertility and not mention Y-chromosome microdeletions and duplications. So there's this 10 megabase pair region on the long arm of the Y chromosome that contains these regions defined as the AZF-A, AZF-B, and AZF-C regions. And so those contain a number of genes that are required for sperm production. And so without those, then male would be azoospermic. And there's clinical relevance in knowing which regions contain microduplications or deletions, because then it would inform the clinician of whether or not a TESI, which is a testicular biopsy, would be effective at finding sperm and potentially using it for assisted reproductive technology. And so again, monogenic causes. In one of the last slides, I think I'm going to show you, I'll talk more about the monogenic causes. So there's post-testicular causes. And so primarily, the CFTR gene that causes cystic fibrosis, that one is interesting because what it can do, besides, of course, all of the various pulmonary issues and other issues that men with CF have, it can also cause bilateral or unilateral absence of the vas deferens. And as you can imagine, without a vas deferens, then sperm have nowhere to go. It's the exact same thing as having a acquired vasectomy. And so CF mutations can do that as well. And then besides loss of one or both of the vas deferens, there's also strange abnormalities that can occur in the epididymis, the organ that helps to mature sperm, that would lead to epididymal obstruction. Okay? So here are the guidelines according to the AUA, the American Urological Association, and the American Society for Reproductive Medicine. So this is what they say in terms of what is recommended for a clinician to test for when a man presents to the clinic with suspected male infertility. And so karyotyping, that one is recommended, of course, because of the XXY syndrome, XX male syndrome, other chromosomal abnormalities that can lead to male infertility. Y-chromosome microdeletion testing, which has historically been a multiplex PCR reaction in order to look for the presence or absence of various loci that would indicate that there are regions that are deleted in the AZFA, B, and C. And then lastly, CFTR testing to look for single or biallelic mutations. Now, what's interesting here is that all the other genes that could underlie male infertility are not included here. So this is obviously current guidelines, and so in the future, it's likely going to update and include many of the genes that are definitively linked, of which there are a good number. Now, interestingly, we are lagging behind the mouse. This is what I've found when I've looked at the MGI, the Mouse Genome Informatics site, in terms of what we know and what we don't know about genes and their requirement for male fertility. So there's obviously a lot of genes that have been knocked out and that have been elucidated to be required for fertility, at least in the male. And so there's still some genes that we're lagging behind that haven't been tested. We haven't ablated it yet. And so we don't know. But then in terms of what we do know for men, what is definitively linked, all right? And so there's a fine line there because there's suspected and definitively linked. The number that are definitively linked is actually quite lower. It's about 117 as of like a year or two ago, and I'm sure the number has grown, but it's still less than 200, probably less than 150 genes that are definitively linked. And so part of the research that I do is to try to increase that number, to try to show cause and effect, and to try to show that variants identified in some of the men that I sequence help to support the notion that it is required in men as well. And so the requisite slide that shows the methods, the bulk of it is the Epitome human workflow platform that does CNVs, SVs, and SNVs. That's huge. It takes a lot of the burden out of the analysis, so then I can focus on the annotation, variant filtering, prioritization, and looking at the variants from the lens of the ACMG guidelines. So you see at the bottom, there's a number of pathogenicity prediction tools that I implement. And those are important to help guide me in terms of what is relevant and what's not in order to make an assessment. And so like I said, there's a lot that happens after the bioinformatics has been done that's still required. And that's also what I wanted to focus on for this talk, is basically the amount of work that has to go into being able to find clinically relevant variants, to be able to report those. And so it is an extensive amount of work, and it's significant, but nanopore sequencing helps with the bulk of it. And also, what I failed to mention previously was that the whole point of this is to show how just through long-read sequencing, we can consolidate multiple tests, right? Karyotyping, multiplex PCR for Y-chromosome microdeletions, SNVs, and then structural variants, which we couldn't have seen before through short-read sequencing. Okay? So some cases, and this one's a pretesticular cause, but it's not Kallmann syndrome. And so this is a 27-year-old male who he was 27 years old when I sequenced his sample, but he had initially presented to the clinic in his early 20s. And when he initially presented, he had failed to have gone through puberty. His LH, luteinizing hormone, which is secreted by the pituitary gland, was basically undetectable. And his testosterone level, which if you don't know what the normal range is for men, it's 300 to 1,000. And so his was less than 20. And these are not his exact numbers, they're approximations, so that helps to de-identify him. And so basically, he obviously has a pretesticular cause. In fact, the clinician had ordered a brain scan to see what was going on in his pituitary. And so I know that the karyotyping had already been done, Y-chromosome analysis had already been done. And so what I found right off the bat was this homozygous LHB mutation, which would introduce this frame shift, as you could see at the bottom. I wish I had a laser pointer, but you could see at the bottom what the native protein would look like. It's a secreted protein with a signal peptide. It's a rather small one. Of course, it acts as a hormone, but the one at the bottom shows the degenerate form because of the frame shift and premature stop codon. All right, so this is a pretty, it wasn't curated in ClinVar, but it wouldn't necessarily have to be, and stated as pathogenic for it to be pathogenic because of the nature of this mutation, where it would completely disrupt protein function. And it helps explain, it perfectly explains why his LH was undetectable. And I've performed all of these, I performed Sanger sequencing confirming. Next case, 24-year-old male, azoospermia, delayed puberty, similar to the previous individual, but with short stature. And so I actually right away found this variant in growth hormone receptor, but then I had to dismiss the variant because ClinVar said it was benign. So I spent a really long time, I believe it was like 2 days, looking at his results, trying to figure out what else it could have been that caused his phenotype. And that's when I went back to the literature. I was like, it has to be the growth hormone receptor mutation. Went back to the literature and actually found it had been knocked in to a cell line, that very mutation, which showed constitutive activation of the receptor, which would cause growth hormone insufficiency. So what was annotated in ClinVar was incorrect. And what was annotated in ClinVar further was that that amino acid position is not conserved, which is not true either. So there's a little bit of pushback. A lot of pathogenicity prediction tools showed that it was pathogenic. And so in this case, I think this is worth reporting. Another one, 35-year-old male, azoospermia, cancer-like lesions in the testicles. And so this one's interesting because the clinicians had informed me about this patient, and a particular note, they wanted me to know that they had felt and seen through ultrasound these lesions, which they thought were cancer. So whatever is causing his infertility could also be causing some sort of cancer phenotype. And so again, karyotype normal, Y chromosome normal. Here's what I found. MSH5 is a DNA repair gene, and he had biallelic mutations. The first one is interesting because it wiped out, it was a splice variant, wipes out an exon that wipes out 21 amino acids. So it's still in frame, interestingly, but every one of those amino acids in the exact configuration completely conserved across 100 species that I checked. So that's interesting. So that variant's pretty significant. The other one, also many pathogenicity prediction tools showed that it was significant. But here's another case where I had to argue against ClinVar because it says it's benign for spermatogenic failure, but pathogenic for ovarian. But the process of meiosis in males and females, that particular process, is pretty conserved. And so it's likely to be an annotation error in ClinVar. And the knockout mice, both males and females, are infertile with defects. So that helps explain this guy's. Now, I've shown the monogenic causes, but I want to show you how, just right out of the box, you guys may already be familiar with this report, right? It's the C&V report. And at the top already, it shows what the karyotype is of the patient, which chromosomes have been detected as 1 or 2 or none. And so this is what a normal male looks like, right? Just as XY, the QD and AC plot shows 2 copies of every chromosome for chromosomes 1 through 22, and only 1 for X and 1 for Y, as you can see to the far right. Okay? 2 copies, 1 copy, 1 copy. This is what a female looks like, right? And so 2 copies of X to the far right, no copies of Y. And then I ran a few Klinefelter's guys, knowing they had Klinefelters, just so that I could see this. There you go, XXY. The QD-seq plot looks exactly like you would expect it, 2 X's, 1 Y. And there was this one individual who, when the clinician sent it to me, I intended to use it as a control for Y-chromosome microdeletions, because when they ran the Y-chromosome microdeletion test through a clinical lab, that's what they found was deletion of many of the genes within the AZF-ABC region. But what was interesting was this showed he had no Y, and which was an unexpected result. So I actually went on this hunt looking for SRY. And the reason being, like I said, without SRY, he would not be phenotypically male. And so he had to have SRY somewhere. And that's when I went back and I found he actually just lost the entire long arm of the Y chromosome, and he still had the short arm, which is pretty cool. And so I guess you could say the QD-seq data will take you pretty far, but then there's a little bit left in cases like this. And so lastly, this is post-testicular. In just the 2 weeks since I submitted these slides, and today, I had an even more interesting case of another male in his 30s, completely undiagnosed for cystic fibrosis, but with a childhood history of pulmonary diseases. And he had biallelic mutations, the Delta 508, and another one that's pretty severe. This guy, he's got, and that guy, he had a unilateral absence of the vas deferens. This guy, it's epididymal obstruction, and then he's got this one curated as pathogenic variant in the CF gene. And so conclusions are that what we all know, basically, is that long-read sequencing is highly informative, and we could potentially, I believe, replace many tests with just performing whole human genome sequencing with long-read. I'd like to thank my co-investigators and the members of my lab for helping us support all the work.